cold case solved, and what you need to know about the newish tool NGS. NGS, Next Generation Sequencing, is a cheaper and faster way to catalogue the entire genome of an animal, plant, or microorganism. Although it's been possible to discover the DNA and RNA sequences of organisms for decades, it used to be prohibitively expensive. NGS methods are much cheaper than older technologies, and this means that it's now practical to use DNA and RNA sequencing to discover which strains of a pathogen are responsible for illnesses. I'm going to quote here from some researchers in a book. The human genome took almost 13 years to sequence in the 1990s at a cost greater than $1 million, whereas now it is possible to sequence the human genome within a week at a cost of $100. When a foodborne illness outbreak occurs, investigators attempt to link contaminated food with samples from sick people. They look for similarities between the organisms in the food and in the blood or stool samples of victims. They don't just look for species-level similarities, but for strains of the microorganism. For example, Listeria monocytogenes is a species of bacteria, one of 17-ish species that belong to the genus Listeria. Among the 17-ish species of Listeria, only 2 are known to be pathogenic, including the deadly foodborne pathogen Listeria monocytogenes. At the species level, there is quite a lot of variation. For example, L. mono, as I like to call it, has at least 14 known strains. Think of a strain, more properly called a serotype, as you would a breed of dog. All dogs are the same species, but Dalmatians are different to Chihuahuas, for example. Identifying the strain of a pathogen allows for better tracking of infection sources. For example, if you find the same strain of Listeria monocytogenes in a food factory and a sick person, you've got yourself more of a smoking gun in epidemiological terms than finding unrelated strains. Until recently, if you wanted to find the strain of a bacterium, you had to perform a series of chemical tests on a bacterial culture growing on agar plates. Different strains respond to different chemicals and dyes differently, and so the results of a series of chemical tests were needed to identify a strain with confidence. With NGS, you can accurately match bacterial strains in food, factories, and patient pathology samples quickly and with a high degree of confidence, and at a reasonable cost. The power of NGS. A mysterious series of sporadic listeriosis illnesses was solved using NGS this year. The illness had been appearing in Bavaria, Germany, for years. More than a dozen people had become seriously ill and 1 person had died. Epidemiologists had not been able to determine the source of the illnesses because it is difficult to know which foods they might all have eaten. Listeriosis has a long incubation period. It averages 21 days and can be as long as 70 days. Imagine trying to recall every food you had eaten in the past 70 days. Then, in 2020, researchers who were checking historical Listeria samples from previously sick patients discovered a link between 13 patients. The patients were all from Bavaria and had all been sick with listeriosis in the past decade. The researchers were using NGS to determine the genetic sequence of the bacteria. They found almost identical genomes in these linked patients, which means the bacteria came from the same source. This detailed information about the exact serotype of the pathogen allowed Bavarian food safety officials to discover the probable source of all 13 illnesses. They detected an exact match of the bacterium at a food company that was supplying canteens and care homes with fruit and vegetable products. This exact match genetic sequence was the smoking gun that linked the produce company to the illnesses and death. Takeaways. NGS technology makes it easier for investigators to pinpoint the source of foodborne illness outbreaks, including old cold cases and cases that might not initially appear to be linked. This makes it more probable that food companies will be caught if they supply contaminated food. In short, NGS is next-generation sequencing. NGS is a cost-effective way to sequence the genomes of food pathogens. Researchers used NGS to sequence Listeria monocytogene strains from patients in Bavaria, Germany. There had been a number of listeriosis cases in Bavaria for which a source had not been found using traditional methods. NGS allowed investigators to match the genetic sequence in patients and environmental samples from a produce company. NGS makes it easier for investigators to link food companies with illnesses compared to traditional microbiological methods. And the sources are in the email and the post from this week's issue.